Hi, my name's Doreen Sakamoto from Amgen. I've been working in the inflammation oncology department for about 14 years now. I grew up in the Central Coast near Morro Bay, and I went to UC Davis where I studied zoology. After I got my degree, I went to Kenya for 2 years of Peace Corps work. I taught biology and math in East Africa, and I met wonderful people. I saw beautiful savanna and really amazing animals, but at the same time, I saw a lot of people who suffered from malaria and from HIV/AIDS and hepatitis. And when I came back, I felt like I really want to study infectious disease. So I was very lucky, and I studied under a wonderful scientist by the name of Kathy Steimer at Chiron. I worked in the HIV project. I've also worked on hepatitis as well as hepatitis C. And I did some oncology work at Chiron. I also worked at Northwestern University and studied molecular biology or HIV molecular biology. And now I'm here at Amgen. And I've been in this department for quite a number of years. And way back when I first started working in the lab, one of the first things I learned was tissue culture. So that is in the beginning of this session. We'll show you a couple slides or a couple videos, one in English, hosted by my friend Sumi, and one in Spanish by my friend Carlos. And they're about 6 to 7 minutes a piece. And afterwards, I have my buddies from the lab, and they will introduce themselves, and we'll have a Q&A session. So if you have any questions, even now, you can start typing them up, and we'll answer them one by one as much as possible. So with no further ado, I'm going to start the introduction to tissue culture, and this is Sumi Roy. So here we go.
Hi everyone, my name is Sumi Roy and I work at Amgen in the oncology department. Today I'll be showing you some basics of cell culture techniques. So when we're working in vitro, which is in a glass dish outside of the tissue's environment, we can grow them in different types of flasks. And we have some primary cell cultures, for example, cells from the bone marrow growing in a 6-well plate, or we have cells in suspension growing in a small flask, or we have adherent cells that are growing in a bigger flask, and we will maintain them in culture for our experiments. So in order to keep our cells healthy and happy, we grow them in this liquid called media. It's not delicious, do not drink it. We have our basic cell culture media, and we add a variety of components to it that it needs for its healthy growth. That includes serum with growth factors. We have essential amino acids, glutamine, and then to prevent anything else from growing in the cell culture, we have antibiotics. So we grow our cells in these incubators where the environment is very regulated to make sure that, again, the cells are happy and healthy. And that includes growing them at a temperature of 37 degrees and 5% CO2. And you can see that we have all our cells growing in here in flasks. And I'm going to grab our cells of interest. And then these are adherent cells. So they are stuck to the bottom of the flask. And in order to continue growing them, I will remove them from the flask and then grow them in a bigger flask. So right next to our incubator, we have our laminar flow hood that again controls the environment and keeps it clean as we work with our cells. That is very important. So we make sure that everything is clean and the airflow inside is being regulated. So I spray it down with a 70% ethanol bottle to make sure everything is sterile. I will wipe this down. And we can start working. So as I mentioned, these are adherent cells and we need to lift them off the surface of the flask. So the first step is to remove the media, and then we'll add trypsin, which does an enzymatic digestion and lifts off the attachments from the cell. we are adding trypsin. And then we'll let the enzyme do its thing in the incubator. So it's been about 2 minutes and we'll take the flask out of the incubator. The cells are now in the solution and we need to neutralize the trypsin with some media. So I'll grab some media. Now that the cells are in suspension with the trypsin, we need to neutralize the trypsin with adding media and then we'll spin down the cells. So this is the media that we prepared with all the components we discussed. And let's wash the flask with the media. So now we'll transfer all of the cells with the media into this 15-man conical so that we can spin it down. So now we'll centrifuge the cells, and I'm balancing the centrifuge with an equal volume, and we'll spin them down for 5 minutes. Okay, so after 5 minutes our spin is done and we can look at the cells, taking it out from the centrifuge. As you can see visually, there is a cell pellet and it's separated from the media. So now we can remove this media and replace it with fresh media and then grow it in a different flask. So we really want to find out if the changes we made to the cell actually worked or not by overexpressing certain proteins. And one way to do that is through fluorescence, and we can find out through this microscope. You can see the image of the cell here, and we want to see if our overexpression worked through fluorescence, and you can see that in red here, and voila, it worked.
So this is the cell. It's made by Beckman, and what they're doing is they're counting the cells. It's a machine that will take in your sample, it will mix it with something called trypan blue, and then the trypan goes into dead cells, and then they put it across 2 thin layers of glass, and then they have a camera and a detector which will detect the cells that have taken up trypan blue and the ones that are bright. The ones that are bright are circled in green, and that will be counted as a live cell, and the ones that are circled in red that has a lot of dark color in it is considered a dead cell. And then they count them, they do the math, and they count and tell us that if what the viability is, and also how many viable cells per milliliter we have in our suspension.
Alright, so now we went through this process of tissue culture, and you might be wondering why we went through all this effort. So we actually do this on a very regular basis to test our drugs in vitro and in a way to see if they would work in the clinic. But in general, tissue culture practices have been used for vaccine development, sequencing of the human genome, and many other useful applications. So I thank you for your attention, and take care. Thank you so much.
So I will share one more video with you.
Hola, ¿cómo están? Mi nombre es Carlos Briseño, soy investigador en el Departamento de Oncología de Amgen y hoy les vamos a platicar sobre una de las técnicas más cotidianas y más importantes que utilizamos, que es el cultivo celular. También les vamos a mostrar el laboratorio donde trabajamos y vamos a hacer un poco de actividades con ustedes. Bueno, y aquí tenemos diferentes tipos de cultivos celulares que, como pueden ver, crecen en diferentes tamaños y aparte son diferentes tipos de células. Por ejemplo, en esta placa de 6 pocillos tenemos células primarias de médula ósea. En este contenedor pequeño tenemos células que crecen en solución, y en este contenedor un poco más grande tenemos células que crecen adherentes. Entonces, dependiendo del tipo de célula y dependiendo del tipo de experimento que queremos, es el tamaño del contenedor que vamos a tener nuestras células. Para mantener nuestras células vivas, las tenemos en una solución, un medio de cultivo. En especial porque muchas de nuestras células son células inmortales, que crecen indefinidamente y que continúan creciendo y dividiéndose. Entonces las tenemos en una solución como esta, que es una mezcla de diferentes cosas, como el medio, el suero bovino que trae todos los factores de crecimiento, algunos aminoácidos, azúcares y aparte antibióticos para evitar que crezcan bacterias o algunos otros gérmenes. Entonces con esto es como podemos mantener nuestras células vivas mientras las cultivamos. Estas son las incubadoras donde tenemos nuestros cultivos celulares. Aquí es donde nuestras células crecen y se desarrollan. Las mantenemos a 36-37 grados centígrados con 5% de dióxido de carbono para mantenerlas felices. Y aquí tenemos diferentes tipos de células. Y estas son las células que el día de hoy vamos a pasar a nuevos contenedores. Esta es nuestra cabina de flujo donde hacemos todo nuestro trabajo de cultivo y la vamos a prender y la vamos a limpiar con alcohol para estar listos para poder hacer nuestro trabajo del día de hoy. Ahora que ya hemos limpiado nuestra cabina de flujo, podemos ir por nuestras células que están en la incubadora, transferirlas a nuestro lugar de trabajo y poder cambiarlas de este contenedor a uno nuevo. Lo primero que hacemos es aspirar el medio de cultivo que ya está usado, y esto es fácil con esta probeta. Vamos a aspirar este cultivo en donde tenemos nuestras células. Ya que hemos removido el medio de cultivo que está utilizado, ahora vamos a poner un poco de tripsina, que es una enzima peptidasa que nos va a ayudar a despegar nuestras células del contenedor donde crecen. Para poder activar esto, estas células van de regreso a la incubadora por un tiempo de 5 minutos. Nuestras células ya están listas para poderlas pasar en la centrífuga. Entonces las incubamos con tripsina para poderlas despegar del contenedor, y como pueden ver ya estamos listos para pasarlas a un tubo. Bueno, y ahora vamos a detener la reacción de la tripsina agregando medio de cultivo. Y vamos a transferir nuestras células a un tubo cónico para después centrifugarlas y poderles así cambiar el medio de cultivo para poderlas pasar a un nuevo contenedor y así ponerlas de regreso en la incubadora. Nuestras células ya están listas y como pueden ver apenas se ven poquitas aquí, pero ya están sedimentadas y estamos listos para el siguiente paso. Bueno, Y ahora vamos a aspirar el medio que ya está usado. Y vamos a resuspender estas células con medio nuevo. Y vamos a transferir una quinta parte o el 20% de estas células a otro contenedor que tiene medio nuevo, y así poderles dar espacio para que crezcan y comida fresca para que se puedan desarrollar. Y ahora que ya está en nuestro nuevo contenedor, le regreso a la incubadora para que nuestras células puedan crecer, y en 2 días vamos de regreso a poderlas utilizar en nuestro nuevo experimento. Una de las cosas que podemos hacer es modificar nuestras células para que expresen algunas proteínas que nosotros queramos. Por ejemplo, aquí tengo células que parecen normales, pero cuando vemos por un color fluorescente, como en este caso es el color rojo, podemos saber si están expresando nuestra proteína de interés. Y como pueden ver aquí, tenemos muchas células que son rojas y que las hemos hecho fluorescentes. Bueno, ya que hemos terminado de presentar lo que es el cultivo celular, se van a preguntar, bueno, ¿y esto qué tiene que ver? El cultivo celular ha sido muy importante porque hemos descubierto vacunas, diferentes medicamentos, e incluso hemos podido secuenciar el genoma humano utilizando el cultivo celular. Y nosotros aquí en Amgen utilizamos estas células para poder experimentar con nuestros medicamentos y asegurarnos que puedan ser eficaces y seguros. Pero bueno, muchas gracias por su atención y espero que hayan disfrutado de este video. Hasta luego.
Carlos, you did a great job. So right now, I'm going to introduce everyone one by one. They're going to tell you where they're from and what they studied, where they studied, and their interest in science. So we'll start with RC.
Hello, everyone. This is RC. I'm from Taiwan, and 7 years ago, I came to the state and get my graduate training in Washington University in St. Louis. And right now, I study the blood vessel in a tumor. Thank you.
Cody? Cody, you need to unmute. Sorry.
Thank you for stopping me. So my name's Cody. I'm from Raleigh, North Carolina, and I studied at UNCW, where I got a bachelor's in marine biology and chemistry. And then I moved to Merck, where we actually manufactured the chickenpox vaccine using cell culture. And I worked in quality assurance there, making sure that the vaccine was safe. And then I went to graduate school where I got a degree in immunology and microbiology with a focus in autoimmune disease and specifically multiple sclerosis. So thank you.
Wing?
Hi, everybody. I'm Wing Lam. I was born in Hong Kong, but grew up in the Los Angeles area. I did my undergrad at Carleton College in Northfield, Minnesota, and then continued on to Washington University in St. Louis, where I did my PhD in immunology. After that, I've come back to California, and I'm doing my postdoc, and I've been at Amgen for about 3 years studying T-cell biology within the tumor microenvironment. Thanks for coming.
Annette?
Hi, everyone. My name is Lynette Chia. I'm from Long Beach, California. I went to UCLA for my bachelor's and then to Atlanta at Emory University for my PhD in immunology. There I focused on vaccine development and infectious diseases like HIV specifically. And now I've been at Amgen for about 1.5 years working on cancer drug development. Thanks.
And Sumi?
Hi everyone, I'm Sumi. I grew up in New Delhi, India, where I did my bachelor's and master's in biochemical engineering and then bioinformatics. And then I came to the States where in 2012, where I did my PhD at Duke University in immunology. Because I was really fascinated how the immune system can both protect you and harm you in different contexts. And after my graduation, I came to Amgen about almost 3 years ago now. Actually, Carlos and I started the same day at Amgen, so that's a fun tidbit for you. But yeah, since then at Amgen, we get to do a lot of cutting edge research that includes a lot of cell culture, of course, but we work on drugs against cancer. So thank you and welcome.
Carlos?
Hey everyone, I'm Carlos. I was born and raised in Guadalajara, Mexico. Moved to the US 12 years ago for college. Went to the University of Notre Dame and then moved to St. Louis, where I met Wing and went to grad school with Wing at Washington University in St. Louis, where I got my PhD in immunology and then moved to the Bay Area 3 years ago. And here at Amgen, I'm focused on how we can use myeloid cells as a way for a cancer cure.
Ambika?
Hi, my name is Ambika. I'm from India. I did my bachelor's in biotechnology there, and I moved to the US in 2012 for my master's degree in microbiology and immunology. And I've been at Amgen since I graduated almost 5.5 years ago. And here my focus is developing drugs for Department of Inflammation, focusing on lung diseases. Thank you.
Lauren?
Hello, my name is Lauren. I was actually born in Houston, Texas. I grew up in Sacramento, California, and partially Germany. And I went to Boston for my degree in pharmaceutical science at Massachusetts College of Pharmacy and Health Sciences, now university, very long name, but they specialize in only health science degrees. I then got my first job at Harvard Medical School, and I was there for about 4.5 years. While I was there, I took advantage of the benefits in order to become a graduate candidate in biotechnology management. From there, I worked at a startup, also doing immunology research. And then I worked at Jackson Labs, which was a contract research organization carrying out experiments for clients such as AstraZeneca, any of the big names that you can think of. And then finally, I landed here happily at Amgen, and I've been here for about almost 2 years now, and I've loved every day of it.
Thanks, Lauren. Prachi?
Hi everyone, my name is Prachi and I'm from Bowie, Maryland. I went to the University of Maryland where I got degrees in biology and English. In college, I worked on a bacterium that infects blueberries and other plants. I then spent 1 year at the National Institutes of Health working on cancer T-cell therapies before I went to Washington University in St. Louis, where I met Wing, Carlos, and RC to get a PhD in immunology. I worked on how cells that are great at fighting cancer can actually develop in your body. And now I've been at Amgen for about a year now, and I'm studying neuroinflammation.
Thanks, Prachi. Christy?
Hi everyone, I'm Christy Perez. I grew up in Menlo Park in the peninsula, and I went to college at UC Santa Barbara where I studied biology. Then I worked in a few different labs and eventually wanted to work on making medicines for patients. And so eventually found my way at Amgen, where I'm studying how the immune system fights cancer.
Great. Thanks, Christy. Thanks, everyone. So we'll go to our question and answer. There's 12 questions that I see. First, what is Amgen? A lab, research facility, or something else? Cody, why don't you answer this one?
All right, I'll turn off my mute this time. So yeah, Amgen is a biotechnology, pharmaceutical company that has many labs, many manufacturing plants, many doctors, and brings pretty much all those things together to both design, develop, and then manufacture, and then deliver those drugs to patients. So I guess it's a mix of all of those, but mainly it's a biotech company.
Yeah, and I should add, I didn't mention this before, but we not only work on inflammation diseases like Crohn's disease, but also, oncology, which is cancer, but metabolic disease such as diabetes, as well as cardiovascular diseases. So we try to hit all the major grievous diseases. We try to find medicines that combat it and alleviate suffering. So the next question is, how many people are here? So we have multiple sites. Maybe I should answer this real quick. We have multiple sites. We have one here, Thousand Oaks, which is our main campus, and Cambridge, Massachusetts. And we have a smaller site in British Columbia and in Germany. Actually, we have many, many sites, but altogether, I think about 16,000 people. Does anyone want to add to that?
I think that sounds about right. Definitely a lot of people around the world.
Yeah, it's a multinational company. We have manufacturing sites in Puerto Rico. We have a site in China, in Japan, not research sites, but the major research sites are here, and Germany and Canada. But I could be mistaken. We have many, many sites. It says, what is this video about? So that's the third question. RC, you want to answer that one?
Sure. So this video is trying to deliver the idea like how we get involved into science and what we really do in our daily life, like working as an employee at Amgen, or like what the potential you can be if you just join us to study biology in the future. Yeah, that's kind of the message we want to deliver here, and also trying to inspire you about what we are doing here. If this could fit with your goal for your career development, then you can start thinking about science and you can take that practice in your daily life too.
Thanks, RC. So Carol asked a question. I took a CCSF forensics class where we learned that DNA replication can sometimes lead to errors in sequencing. Please comment, especially on how often that type of error occurs. Let's see who would be able to answer that. Who's the molecular here? Lynette? Prachi? Can you guys?
I guess I can answer this one if
Okay.
So routinely, the body is always trying to find any errors that are happening. So it's more about how prevalent the phenotype ends up being from these DNA mismatches or RNA. And typically, there are genetic disorders that are caused from those mismatches, and those will end up becoming some type of indication of a disease or mutation that becomes either a physical manifestation. And so that is where there will be treatment that comes in to either help those people get better or at least maintain a healthy status in their life. But DNA, there's generally like a lot of things that our body is really good at fixing because those things do happen.
Thanks, Lauren. I should just add that our cancer is basically our own cells that have gone awry, and usually it's our immune system that helps identify those crazy cells and gets rid of them. But sometimes it that cancer cell can evade our immune system, and then they start growing and they become a tumor. So our job here, one of our jobs in inflammation and oncology is actually to harness our own immune system to attack these cells, these tumor cells. And that's the new frontier actually in oncology. Let me see. The next question is from Ade. I'm sorry if I mispronounced your name. Is there a specific air content or pressure needed inside the flask? Sumi, you wanna go for that one?
Sure. I know the conditions in the incubator have to be very tightly regulated. I think it's about 1 bar of pressure, and Carlos can correct me if I'm wrong because I know he pointed it out in his video. But the temperature, the CO2, and the pressure, everything is very tightly controlled in the incubator.
Yeah, so air pressure is pretty much the same as what we experience out here in the incubator. The only thing that changes is we're pumping carbon dioxide into the incubator so our cells a little bit happier. So we keep that at 5% as opposed to what's normal in the atmosphere, which I can't remember off the top of my head. But yeah, but other than that, like the oxygen levels are the same, nitrogen is the same. Wing, however, works on some models that are hypoxic, which means that she keeps the cells in lower oxygen. So that way, it's a good way to study how those cells respond to low oxygen conditions, like in a tumor, for example.
Yeah, kind of building off of what Carlos was saying. So our because it's the same, so O2 normally is about 20%, and I have a special incubator that pumps in nitrogen and pushes out oxygen. And so my cells are maintained at 0.5% oxygen, and that's kind of one big difference. And the reason I'm studying that is because we know that in a lot of tumors, the cells can experience oxygen at low levels, like around 0.5% or even lower. So yeah, thanks.
I can just add something really quick since he specifically asked inside the flask. So the flask caps are actually vented, so the air can actually go out and come out, and they're not airtight. So the temperature and everything inside the incubator is also maintained inside the flask.
And the temperature is 37 degrees Celsius, which is the same temperature as your body. So that way the cells are growing in a condition that would mimic what's happening in your own body.
Okay, so this question comes from Samir, and the question is, are there protocols in the lab that change due to COVID-19? Lynette, you want to take that one?
Yeah, I can take that. So some protocols that have changed in lab due to social distancing measures that we've implemented to keep all of our researchers on site safe include, not having too many people in one area in those tissue culture hood areas that you saw the videos of. So there's only up to 3 in basically one area that we're allowed at any time. And then even in our office space where we have kind of a shared kind of desk situation, we've kind of made it so it's kind of every other seat that's available to make sure that we're kind of separate. And we also try to utilize like offices more so that we're pretty separate from each other. We have some groups have sign-up systems to make sure that when we're trying to use instruments or rooms for experiments that we're kind of scheduling ourselves so that we're not all around each other at the same time. So it kind of works out that way.
Thanks, Lynette. And you'll notice that everyone's not wearing a mask, and I asked them not to wear one in case you need to read lips, but we normally have masks on if we're with each other. So everyone is in their separate rooms answering these questions.
So actually, if you notice in the video, Sumi and I were wearing masks. That's just because it was a normal work day. That's the reason why we're wearing masks in the video, because we're in the lab, and now that we're out here, we're all in our own room. So that's why we're maskless, at least for a little bit.
The next question is, what is the red chemical that the cells grow in? Why do we grow cells in that chemical rather than water?
I can take that.
Okay.
Hey, so the red chemical that the cells grow in is media, so cellular media, and the point is to try to mimic the conditions that the cells were growing in as closely as possible. So the red media has like a bunch of vitamins and salts and proteins, and as Carlos and Sumi showed you, they added in like some antibiotics so that the cells don't get contaminated. But we don't grow cells in water because the cells would die, because the just the balance of vitamins and plasma would be off.
And the osmotic pressure, if you guys remember that. Next question, do you think we could make—
Actually, can I add something?
Sure.
So, and then it's red because the red is actually a pH sensor. So if the pH, once the cells start eating up all the vitamins in the media, they actually start releasing acidic content. And so our media turns yellow, and that's a way to tell us scientists that like the cells have eaten everything and it's time to give them fresh media. So whenever we come in and our media is really yellow, it's worrisome. It's time to feed them, and that's how we keep them happy. So red means actually good. And there's a question right now in the chat saying can media affect the outcome of the cell growth? And yeah, it depends on what type of media we're using, all the other additives that we have. So we can give it different proteins to make them grow quicker or to slow them down a little bit. Anyway, sorry.
No. Thanks, guys. Perfect. So someone asked, do you think we can make artificial eco-friendly meat in the future?
Isn't that what impossible meat is? I think it's like grown in a cell culture.
Yes,
So
it is.
I guess that's up to your own opinion. I have no idea. It seems like a lot of resources, especially lab resources, they're definitely not cheap, but I think alternative to how much methane gas cows produce, it is more economic for the environment.
Thanks, Lauren. So Samir asks, how does growing these cells, these tissues and cells work? How can you use this? Does anybody want to take that? Cody?
Yeah, so you can get a lot of useful information from cells. So in general, it would start with what cell or tissue you're interested in. So for example, we can grow cells from a lot of different tissues like the liver, the spleen, the kidneys, the lymph nodes, the thymus, the list goes on. And then you can also grow cells from a lot of different animals. So you can grow them from insects, you can grow them from fish, you can grow them from mice, people, humans, rats. And so that allows us to ask a lot of different questions. So we can learn about different processes within a single cell. We can learn about processes of how cells communicate with each other. And we can ask how our drugs affect the cell, so how they actually change how the cell behaves. And that's most what we're interested in, is when we apply a drug or a medicine, as we call them, what does the cell do to respond? And that can be measured through a lot of different outputs by either the cell's death, the cell's growth and survival, the way it looks physically, or we can actually program them to give us a sign that they're happy or not by the expression of green or red or yellow proteins.
Thanks, Cody. So we have a CRISPR question. You know, as you know, the two women who discovered CRISPR won the Nobel Prize this year. And so Ave asked, do you use this with CRISPR biotechnology to help create cells designed to fight cancer? Good question. It's done CRISPR here. Not Carlos.
Yeah, so actually, so we use CRISPR to make changes in cells to get a better understanding of how those cells work. So will they respond better or worse to one of our medicines, for example? And how is our medicine how does it work inside a cell? And so we use CRISPR for that. And then another way we use CRISPR is if we're interested in one particular gene, we can just take our T cells that we culture, so target them with CRISPR, and then we can figure out if that gene we got rid of was important or not for a T cell response. And so in the bigger scope, whereas while we're not coming in with T cells taking CRISPR and then sticking them back in humans, we're using CRISPR to understand how we can make better medicines, how we can make better therapeutics. That's a way to fight disease. So it might not be direct to cure cancer, but it's a very cool tool, and as Doreen commented, very rightfully won the Nobel Prize this year, which is super quick. I mean, if you think about it, like CRISPR, the boom was like 10, 15 years ago.
Thanks, Carlos. Joseph asked, do you guys work with animal cells, plant cells? I think we answered that, but we don't work with plant cells, we work with animal cells. And insect cells, actually, as well. Someone else asked, what were the liquids that went into the flask before putting it into the incubator? I think we answered that, but you might have come in later, so maybe Sumi can answer that real quick.
Yeah, so as Prati covered in an earlier question, and also as it's shown in the video, the liquids essentially have all the nutrients and ingredients that are essential to not only keep the cells that we are growing out of its own system in this artificial setup. We will need to give them all they need, amino acids, glutamines, things like that, vitamins that they need for their growth. And it also has a pH indicator in the color, as Carlos mentioned earlier. Thank you.
Thanks.
Are they talking about trypsin?
Yeah, and then the other liquid
Yeah.
we added was
Oh.
trypsin.
So we add trypsin to lift off the cells, the adherent cells. So the trypsin basically breaks the protein that enables the cell to lift off, so we can split them.
And what she means by splitting is just growing them out. So you take a part of them and then grow out, because they just keep on growing. Many of these cells are derived from actual tumors from people. So as you can imagine, they are what we call immortalized. So they keep on growing as long as we feed them. So the next question is Amgen working on finding a cure to the coronavirus? Christy?
Yeah, I think what we're allowed to talk about is that Amgen is interested in figuring out how the immune system works. And so coronavirus affects your immune system. And so we're also researching ways to make sure the immune system doesn't overreact and result in either disease or death.
Thanks, Christy. I do know that Amgen is really good at making antibodies. So we're helping another company make antibodies. So we're not an infectious disease company like Gilead, but we are working on the coronavirus, or a cure or a therapeutic to coronavirus. And that has been advertised.
And we've also supported a lot of relief for first responders and communities by providing masks as well.
Yeah, good point. Thank you, Lauren. Someone asked, where's our main site? It's in Thousand Oaks, California, which is in Southern California near Ventura. Samir asked, what are common tools that you have used every day? RC?
Laptop, of course, and like everything you can see like showing in the video. That's like our daily life. We spend like around 50% of the time in the laboratory to do those tissue culture thing. And then we also spend the other time attending meeting and prepare the notes we did for our experiments.
Yeah, I consider working in the lab as like high-tech cooking. So we have things like, we commonly use dry ice, we have liquid nitrogen, we have regular ice, but we have these small little instruments called pipettes that instead of a cup or a teaspoon, we actually pipette or measure out microliters of material, sometimes less than a microliter. So we can do experiments in a very, very small tube, or we could do in a very large plate. So those are the types of tools we also use. So if you like to cook, it might be something that you might want to do, because it's fun just to mix. It's like mixing things together, and then after a few days, you might have a result. And that's what's so exciting about science, is that we are the first people to see if something worked or didn't work. Next question is, do white blood cells kill cancer cells, or the crazy cells, or both? Who wants to answer that one? Wait.
Sure, yeah. White blood cells, I mean, in the ideal world, your white blood cell can basically recognize anything that is seen as foreign or different. That's kind of how they know what to kill. Typically, you think about that in terms of bacteria and viruses, of course. But yeah, and when tumor cells become tumor cells, they have multiple mutations that they've picked up, and either because of that or as a side effect of that, these cells will also then look different from your normal cells. And yeah, your white blood cells will be able to pick these tumor cells out and be able to kill them. Clearly, it doesn't happen all the time, and that's what here at Amgen we're working on, how to help your immune cells recognize tumor cells as foreign and to then kill them. So yeah, they do.
Thanks, Wayne. Next question. With CRISPR, and if you're able to replicate killer T cells, do you think it's possible to engineer killer T cells to detect cancer? We're sort of doing that right now, but Carlos, since you're our CRISPR person, why don't you take that one?
Yeah, I mean, definitely with CRISPR, you're able to come in, open the DNA, and then you can insert a something into the T cell's genome, so you can express a molecule that could kill the cancer cell. And then engineering the T cells is actually already being used as a therapeutic. So there's something called CAR T cells, in which you express a receptor on the T cell that is going to recognize a cancer cell. And so whenever a T cell finds that cancer cell that binds its receptor, it can then just directly kill that cell. And so that's been really useful in liquid cancers, like lymphomas and leukemias, although solid tumors are a little bit more difficult because not a lot of T cells are getting in, and the solid tumors are also a lot more heterogeneous, or there's many different types of cells in the solid tumors. That's why you don't hear about CAR T cells as much in solid tumors. But definitely, that's one of the more promising approaches right now, is to engineer T cells to kill cancer cells.
Thanks, Carlos. Next question. Do you use bioluminescence in your lab? Who wants to answer that one? Prachi?
Yeah, we do. I think Sumi showed some red cells, and those were made by adding like a virus that has a red protein into the cells, so that the cells that express this virus are red. So that comes in lots of different colors now, like green, which is the original one.
And the person who discovered that actually got the Nobel Prize many, many years ago, but it's been a wonderful tool for all of us to see if something is expressing that we got a gene into a cell, that sort of thing. So in fact, that's what I'm doing today. I'm sorting on cells that have more red in it so that I could isolate a certain population. So Yun Tian Zen, sorry if I butchered your name, but asked, are you guys also trying to find a cure for COVID? So I think we went over that. We are working on antibodies for COVID right now, and we do have antibodies against certain cancers, and we're very good at making antibodies. So so it was Eli Lilly asked us to join with them and make antibodies against COVID. Someone else asked, may we be able to make cells one day? Lynette, you want to try this one?
Sorry, I forgot to unmute myself. I was going to say, we do use cells that we already have access to and generate new versions of those cells. So we're able to, as mentioned before, like add those bioluminescent proteins in them, and that makes them, different than what they were before. And, they can have different characteristics depending on genes that, you either put in or take out or modify in some other ways. So in a sense, in that sense, we are able to generate these different kinds of cells that we're able to work with, and and it's nice because we're able to kind of adapt them to our specific research question, and they're just a very useful tool for just the questions that we ask.
Thanks, Lynette. Next question. How has growing
I think Carlos had something to add.
So there's a guy called Craig Venter who is a multimillionaire, and he started his own institute, and he's trying to make cells from scratch. And as far as I know, I think he got a bacteria cell out, and he published a paper a couple years ago on it. But so yes, maybe as technology evolves, we might be able to make cells from scratch, or human cells from scratch, but we're very far away from that.
Thanks, Carlos. This is a history question. How has growing cell culture developed over the decades? Have we started designing eco-friendly tools like a refrigerator? Instead of using an incubator, use refrigerators because they use so much energy. Is there an alternative? Boy, that's a hard one. I don't think we've ever grown cells less than 37. Sometimes 30 degrees, definitely. We grow bacteria in 30 degrees.
I don't know about eco-friendly, but I know that people used to mouth pipette. So that's just generally safer. Instead of using the cool pipette mans that we have now, people used to literally suck on the pipette to bring like media or liquids up and out.
Yeah,
that doesn't answer the question.
Unsafe. Yeah, it doesn't. Yeah, so I don't think there's an echo. We do use a lot of plastic, and I think.
But at least we recycle at Amgen.
Yeah, we try to recycle everything.
Yeah, like I'd say Amgen has been the most eco-conscious company I've worked for in terms of how they dispose of lab waste. Like we have extra bins, like for the cardboard boxes for them to all go to get recycled. We have for sterile and non-contaminated items that are like our plastic media bottles. Those are all cleaned out and recycled. So I'd say like Amgen specifically does take steps to make sure that we do things as eco-consciously as possible, but it's really hard to limit that when everything has to be sterile and single use in order to maintain a sterile conditions for cell culture.
Thanks, Lauren. We only have 3 more minutes, so we'll try to answer a few more questions. Let me see. This is a good one. What is the difference between mammalian cell culture, cell lines, primary cell culture? Let me see. There's a lot.
So mammalian cell culture are basically cells that are derived from mammals, and mostly we do that to like human mouse. Cell lines are cells that are immortal, and we use those. Primary cell cultures are when we isolate fresh cells from like human blood or mouse blood, and we culture those, but those don't survive as long as like the lines do. If anybody has anything to add.
Sounds good. Okay, I think we only have 1 more question, maybe 2. So someone asked, we just purchased our first microscope for our family to hopefully begin developing an interest in studying life science this year. Do you have any recommendations for what we must see or do with it for those of us just starting out? Let me see. I think I'm the only one with kids, so maybe I should answer that one. Geez, there's all sorts of things. It depends on how good this microscope is, but certainly, you know, looking, getting things from the garden and looking at plant cells is very easy. You could even scrape the inner part of your mouth and get the epithelial cells and put them on a glass slide. You could put and glass slides are very cheap. You should be able to, it should have come with your microscope and put another piece of glass on top of it and look at that. You could look at little bugs on your slide. I mean, there's all sorts of different things. And it's just, I mean, you're very lucky to have a microscope. So, so good luck with that. If you have, I mean, if you definitely online, there's a lot of information about what you can try to do with your microscope, but that's what I would do if I had one at home. Let me see. Let's see if we can do one. Oh, it's 3 o'clock. One more question. Does the media affect the outcome of cell growth? And how do you identify a disease, and how do you know the strength of the disease? Sumi, can you do that one?
Sure. So like people, I think cells have their own preferences. So depending on what you have in the media, they may like it or they may not like it. So they need certain factors to be healthy, which is the base media. But if you make certain changes, say the pH, or say you add something that is not physiologically what it expects, it can go into hyperdrive, or it may slow down the growth. The cells may die off. And your second part is, how do you identify a disease, and how do you know the strength of a disease? I'm not sure what this question means, but we have different cells mimicking the disease state. So we can get tissue, say somebody has lung cancer, we can have lung cancer cell lines, and then we can work on that. And there are different properties of that disease, particularly in cell culture. You can only see how fast the cells are growing, and it affects, if you add a drug, if that's responding or not.
Thanks, Sumi. Thanks, everyone. I think this is the end of our session. And we would love to see you at the Science Festival next year. Hopefully it'll be in person, but please come to the Amgen tent, and likely all of us will be there. Ask us many, many questions, and thank you so much. Thanks, everyone.